Hello and welcome back to Biochemistry for Health Sciences channel. In today's video, we shall talk about the oxygen binding proteins, hemoglobin as well as myoglobin. In previous videos, we emphasized that proteins basically carry out most of the cellular activities. So if we could understand the properties and functions of proteins in each one of the 200 or so different types of cells present in the human body, then we should be able to better understand and appreciate human anatomy as well as human physiology, both in health as well as in disease. Proteins work by binding chemicals that we call ligands. So for example, the proteins known as enzymes that catalyze biochemical reactions, they bind ligands called substrates. Proteins known as receptors bind ligands known as signals. For example, the insulin receptor binds the ligand insulin. Transport proteins bind the ligand, which is the chemical that needs to be transported across the membrane. Antibodies are proteins that protect us against microbes. They bind ligands that are known as antigens. And in today's video, we shall look at hemoglobin and myoglobin, and these proteins bind the ligand oxygen. Myoglobin is a protein found in skeletal muscles as well as cardiac muscles. Hemoglobin, on the other hand, is found everywhere because it is present in red blood cells. The information to make myoglobin is on chromosome 22, while the information to make hemoglobin is on chromosome 16 and chromosome number 11. In terms of size, myoglobin is a small protein, about 17 kilodaltons. Hemoglobin is about 4 times larger. It's about 65 kilodaltons. Clinically, this is important because, as you remember, anything less than 40 kilodaltons can escape into the urine if it is found in the bloodstream in excessive amounts. This means when there is breakage of skeletal muscle cells or cardiac muscle cells, myoglobin could actually come out into the urine, and that is called myoglobinuria. However, hemoglobin is much greater than 40,000. It will not appear in the urine if there is excessive red blood cell breakage. In terms of structure, myoglobin has one polypeptide chain. Therefore, it has a primary structure, the sequence of amino acids in the polypeptide chain, the secondary structure, and tertiary structure. Hemoglobin, on the other hand, has 4 polypeptide chains. These are called alpha 1, alpha 2, beta 1, and beta 2. So each polypeptide chain is also called a subunit. So hemoglobin is basically alpha 2, beta 2. It has a quaternary structure, alpha 2, beta 2. These polypeptide chains in hemoglobin, you can see they are connected to one another by weak forces that we discussed before, such as hydrogen bonding, hydrophobic force, and so on, as well as ionic. So there is ionic or salt bridges or electrostatic interactions between these four subunits to form the quaternary structure, alpha 2, beta 2. Let us look at the active site of myoglobin and hemoglobin. If you recall, the active site is that part of the protein which is actually involved in the function of the protein. So right somewhere inside the protein is the active site of the protein. For myoglobin, it has one polypeptide chain, it will have one active site. While for hemoglobin, there are four polypeptide chains, therefore there will be four active sites. The active site of myoglobin as well as hemoglobin contains a few components. One is an organic molecule that is called heme. Heme is a highly conjugated chemical. You can see there is single, double, single, double, single, double bonds, and so on. And that gives it a very bright red color. So the red color of heme is the reason why myoglobin is sort of brownish red in color and why hemoglobin is red in color. Any problems with the synthesis of heme leads to diseases that we call porphyrias. And we'll talk about porphyrias in future videos. The active site is mostly hydrophobic. It is rich in non-polar amino acids, except for two polar histidines, which are also found at the active site. One is histidine 64, and the other is histidine 93. Histidine 64 is also called the distal histidine. It's present on helix E, so we can call it the E7. Also the E7 histidine. Histidine 93 is called the proximal histidine or the F8 histidine. The active site also contains a transition metal. And the transition metal that is found in both myoglobin as well as hemoglobin is iron. So the Fe2+, as you know, transition metals can bind many atoms at the same time. So this iron atom, for example, can bind the nitrogens of the hemes. So these four nitrogens of the heme. At the same time, the iron can bind the nitrogen of histidine 93. And also, most importantly, this is where oxygen binds. Oxygen binds to the iron atom. So besides oxygen, we could also sometimes see nitric oxide binding to the iron atom or even carbon monoxide. It is interesting to note that carbon monoxide actually binds iron much more than does oxygen. However, the presence of this distal histidine, E7, weakens the interaction between the iron and carbon monoxide. So that's one very important function of this distal histidine E7. The iron atom is not in the same place as the plane of the heme. Actually, because of all these nitrogens here of heme, as well as the nitrogen of the proximal histidine, the heme looks more like a dome-shaped structure, and the iron atom is about 0.6 angstroms out of the heme plane towards the proximal histidine, towards this side. So the iron atom is more on this side, out of plane by about 0.6 angstroms. So this is the basic structure of the active site without oxygen present. So this is the structure of myoglobin without oxygen bound and of hemoglobin without oxygen bound. So we call deoxyhemoglobin without oxygen or deoxymyoglobin without oxygen bound. Now let us see what happens when oxygen binds to the active site. So here are the steps, the essential steps that happen when oxygen binds to the active site of myoglobin or of hemoglobin. In the first step, the oxygen gas will diffuse to the active site. In the second step, when the oxygen is very close to the iron, the electron density around the ferrous ion increases. This increase in electron density around the iron atom causes a rearrangement of the electrons in the d orbitals of iron. Remember, there are 5 d orbitals. So these electrons that are present in the d orbitals of iron, due to the increased electron density, they become rearranged. This rearrangement of electrons in the d orbitals actually decreases the size of the iron atom by about 19%. The iron atom actually becomes smaller when bound to oxygen. The iron atom now moves into the plane of the heme. Since iron is connected to histidine 93, which is on helix F8, this movement of iron atom into the plane of the heme causes the whole helix F8 to also move. And this movement of helix F8 will lead to a change in the shape of myoglobin, as well as in the change in shape of the subunit of hemoglobin. In case of myoglobin, there is only one polypeptide chain, so this change in shape doesn't cause any further dramatic effects. Hemoglobin, on the other hand, has 4 polypeptide chains that are connected to one another. So without the oxygen bound, we call the deoxy state of the subunits, we call it the T state. Remember that these subunits are connected to one another by weak interactions, including electrostatic or ionic or salt bridges. So when the first oxygen molecule binds to one of the polypeptide chains of deoxyhemoglobin, this causes this shape here to change, and the change in the shape leads to destruction of these weak forces such as salt bridges between the other polypeptide chains. This subunit therefore gets transformed from the T state to the R state. So the transformation of the first subunit from the T to the R state causes the second subunit to also become the R state, and more of these electrostatic and weak forces are broken. The third subunit will also become the R state, and eventually the fourth subunit will also become the R state. This means the first oxygen molecule to bind causes the shape in chains of one subunit. So it's harder for this oxygen to bind because this subunit was originally in the T shape. But because of this conversion from T to R state that induced the formation of the R state in other subunits, the second oxygen molecule and the third oxygen molecule and the fourth oxygen molecule will bind to the hemoglobin much easier. So because hemoglobin has a quaternary structure where the four polypeptide chains are joined together, basically the four polypeptide chains are sort of talking to one another, and when the first polypeptide chain changes into the R state, that causes the rest of the chains to also gradually become the R state. And this property of hemoglobin is called the cooperative effect. The cooperative effect, which means gets easier and easier for oxygen to bind to the hemoglobin as one subunit induces the formation of the R state in the other subunits. This is because the R state binds oxygen very well. The T state has a lower affinity for oxygen. To summarize, a comparison between myoglobin and hemoglobin gives us a good idea about the different behaviors of proteins. Those that do not have a quaternary structure, such as myoglobin, do not show cooperative effects, and proteins such as hemoglobin that have quaternary structures can show cooperative effects. The cooperative effect of hemoglobin can be seen very well when we plot the oxygen binding curve, also known as the oxygen dissociation curve. So from the shapes of these curves, we can see that myoglobin does not have a cooperative effect, while hemoglobin does show a cooperative effect. So to get the oxygen binding curve, let's say for myoglobin, what we do is we take many different test tubes. In each test tube, we put a fixed amount of myoglobin, but we increase the concentration of the ligand as we go from the first test tube to the last test tube. So the concentration of oxygen, since oxygen is a gas, we express the concentration as the partial pressure of oxygen in millimeters mercury. So the oxygen concentration is plotted on the x-axis, and then in each tube we measure how much of the myoglobin has oxygen attached to it, what we call as YO2, or percent or fractional saturation of myoglobin, and we plot that on the y-axis. Okay, so basically PO2 on the x-axis and YO2 on the y-axis. And we do the same things for hemoglobin. So here's a comparison of the oxygen binding curves of myoglobin and hemoglobin. This one here is for myoglobin. This curve is a hyperbolic curve. The equation for the curve, for the hyperbolic curve, is shown here. YO2 equals PO2 divided by P50 plus PO2. P50 is obtained by taking 50% saturation, extrapolating it to the curve, and the concentration of oxygen or PO2 here is your P50, and for myoglobin that comes out to be around 2.8 millimeters mercury or 2.8 torr. This is also called the dissociation constant or Kd of myoglobin. So for example, if we want to know what is the percent saturation of myoglobin in muscles, and in muscle cells the PO2 is roughly about, let's say, 40 millimeters mercury. So we can substitute 40 and the P50 in this equation, and we get a fraction of about 0.93 or 93%. That means at 40 millimeters mercury pressure of oxygen, the myoglobin is about 93% saturated. 93% of the myoglobin is bound to oxygen. Now, unlike myoglobin, you can see the curve for hemoglobin looks very different. This curve looks like the letter S, and therefore it's called the sigmoidal curve. And this curve is expressed by this equation, YO2 equals PO2 raised to the power of n divided by P50 raised to the power of n plus PO2 raised to the power of n. Now at 50% hemoglobin saturation, YO2 of 0.5, if we extrapolate that, we get a PO2 here of about 26 millimeters mercury. So this will be the P50 of hemoglobin. You can see this number 26 is almost 10 times greater than 1 for myoglobin. This number n is called the Hill coefficient, and this can be determined from a plot called the Hill plot. In the Hill plot, instead of plotting YO2 on the y-axis, we plot log YO2 divided by 1 minus YO2. And on the x-axis, we plot log of PO2. And when we do that, we get a straight line. And from the regression equation of the straight line, we can get the slope of the straight line. And this slope is equal to n, which is the Hill coefficient. And if we want, we can also get from the x-intercept the P50. So from the Hill plot, if we do that for myoglobin, we will get a value of n equals 1, which means myoglobin doesn't show any non-cooperative it doesn't show any cooperative effect. Basically, myoglobin is a non-cooperative protein, and that makes sense because it doesn't have a quaternary structure. There is no quaternary structure. Hemoglobin, on the other hand, has a Hill coefficient of 3, and that shows hemoglobin is a cooperative protein. It shows a positive cooperative behavior. So knowing N equals 3 and the P50 for hemoglobin is 26, we can once again calculate what is the percent saturation for hemoglobin in muscle tissues, for example, where the PO2 is 40 millimeters mercury. So that would be 40 raised to the power 3 plus divided by 40 raised to the power 3 plus 26 raised to the power 3. And we calculate this, we get about 0.78 or 78%. So unlike myoglobin, where 93% of myoglobin is bound by oxygen at a PO2 of 40, for hemoglobin, only 78% of hemoglobin is bound by oxygen at a PO2 of 40 millimeters mercury. So just to summarize, the P50 for myoglobin is only 2.8 compared to hemoglobin, which is 26. Since myoglobin has a lower P50, that means it has a higher affinity for oxygen. So the lower the P50 value, the higher the affinity for the ligand. Hemoglobin's curve is on the right, which means it also has a higher P50 or lower affinity for the ligand oxygen. Myoglobin's curve is on the left, which means myoglobin has a lower P50 and a higher affinity. Hemoglobin shows cooperative effect where N equals 3, and you can see that from the sigmoidal curve, while myoglobin does not show any cooperative effect, N equals 1, and you can see that from the hyperbolic curve. We will talk more about myoglobin as well as hemoglobin in the next video. Until then, goodbye and have a wonderful day.